So I will go ahead and introduce the first of the last two speakers who will be presenting their talks, I believe, in a pre-recorded manner. I'm not sure about Dr. Bentley if he's with us, but I'll start with Dr. Rana Dajani. Dr. Rana Dajani is an Eisenhower Fellow, professor of genetics at Hashemite University, Jordan, and a visiting professor at Yale and Cambridge. She is also a Cmalakova Fellow at the Jepson School of Leadership at the University of Richmond and Harvard Radcliff Fellow. She received her Ph.D. in molecular cell biology from the University of Iowa in the States. She is also a member of the CAGS Arab Council representing Jordan. The title of her talk will be Who Are We Really? So I hope the team will be able to play her pre-recorded talk seamlessly.
Assalamu alaikum, peace upon you all. I will share my screen so that we can begin our talk. So the title of my lecture today is Who Are We Really? And this is kind of a take on reality because of the ongoing discussion and dilemma, nature versus nurture. So I will share with you the latest research, hypotheses, and theories about this question. But the whole story started with the Syrian crisis. Now, I live in Jordan. I'm half Syrian, half Palestinian. And in March 2011, there was an outpour of a Syrian family and friends fleeing to Jordan in fear for their lives. The majority of these refugees are children. And many international NGOs came in delivering programming to help alleviate the stress and anxiety and trauma that these children were going through. So the question became, which interventions are effectively working to abate violence and promote health? And how do we get robust evidence for program evaluation under humanitarian conditions that exist on the ground? And so I was part of a research project with Yale University, funded by ELRA through Wellcome Trust, to evaluate a program run by Mercy Corps among Syrian refugee adolescents and Jordanian adolescents from vulnerable communities. And we asked the question, what works for whom? Okay, and could we gather high quality and in-depth evidence regarding humanitarian public health programming globally? So this was a program run by Mercy Corps, as I said, called No Lost Generation. It's a psychosocial intervention of 8 weeks, delivering different activities for the adolescents. Now, of course, the program was evaluated in the typical way of mental health surveys and cognitive skills and functions and so on. But we wanted to look under the skin. So the first thing we did was we looked at levels of cortisol by gathering samples from the hair. And we were able to show that with the intervention as a randomized control trial, that the cortisol levels went down in a statistically significant amount, which we published in an article in Psychoneuroendocrinology. However, and these are members of our team who were part of doing this. Of course, what I showed you is just a little bit of the bigger study. However, we were also interested to go beyond just looking at hormones in the blood as one way of peering under the skin. We also wanted to look at genetics. So we know that different types of data give us different types of uncertainty, and the environment impacts human beings. And not just on their physiology, as I said, but also on their DNA and on their genetics. So we started exploring epigenetics. So we already know that cells, quote unquote, listen to what's happening outside, meaning they're impacted by the environment through signaling pathways to impact, in fast responses, protein signals, but in slower responses, DNA expression. right? And so some of this work that initiated a couple of decades ago in mice showed that if you had a pup that was raised by an anxious, low nurturing mother, that pup would grow up to be an anxious adult. And if a pup was raised a DNA identical, but raised by a relaxed, high nurturing mother, then the pup would grow up to be a relaxed, high nurturing rat. So therefore, something was being inherited by the pup through that kind of behavior. And what we found is that this pup we could reverse its behavior by injecting a drug that adds methyl tags. And if you do that, you actually are turning off a gene, and this pup that supposedly was raised by a relaxed, high nurturing mother, instead of growing up to be a relaxed, high nurturing rat, actually becomes an anxious rat. So scientists were able to show that it's the turning off and on of a gene or a number of genes could actually change behavior. And in this particular example of rats, actually the gene that was being turned on and off by methylation was the glucocorticoid receptor that binds to cortisol. And so if you're turned off your gene, you were shutting down the stress response because the receptor was not there. And so the cortisol would remain high. And this really kind of fit into what we were studying in the Syrian refugee children when we were assessing cortisol hormone. So we wanted to look at other genes that may be related to what's going on. Now, how come, for example, the cortisol receptor, the glucocorticoid receptor, is modulated in that way? Because we know usually in evolution, you actually, things that are not beneficial get selected against and eventually disappear. However, this is not being selected against and it does not disappear, meaning that anxious behavior can have an advantage. And that's a similar question when you look at depression. How come it still exists? So there must be some kind of advantage for it. And so shifting to human beings, and looking at the associations between epigenetics and the human brain, and this is very also nascent because of the ethical challenges and barriers of studying brain tissue, but we already know that there are certain epigenetic tags that change and can be related to behavior. And so because of that, we wanted to look at a particular gene and genetic variants that may have an association with the trauma exposure in these children, and to see if there's any connection with stress, mental health, or resilience. And we want to see if the intervention had any impact on that. And the rationale was that the genetic variants may influence risk and resilience shown by the youth with extreme traumatic exposure. And so the underlying rationale of the environmental sensitivity variants, those that were, that you could toggle the knob, that there's a large variation in the human response to negative and positive events. And there's a weak evolutionary argument for genetic variants that only create negative impacts. And we wanted to look also if there were any positive impacts to enhance the individual's sensitivity to multiple environmental impacts, not just negative, but positive. So in the literature, MAOA is a gene that has a repeat polymorphism, and it's an enzyme, and it functions to break down neurotransmitters to inactive metabolites, the neurotransmitters, for example, that MAOA works on is dopamine. And this gene is X-linked, so males only have one copy, and therefore it can be studied in males in a much easier, clearer fashion. And for that particular gene, it has a repeat polymorphism. So it has either 2 or 3 repeats, and those would be described as low activity. And then we have other variants where there's 3.4 to 5 repeats, and that's considered high activity. So in a way, it's like you're toggling, turning off or on something. So it's a nice example to see if compare high activity with low activity of this particular gene and see if there's any correlation with trauma or resilience. And we already know that there are differential associations with sex because it's X-linked. And we know that from other literature that this variant is associated with altered behavior when combined with early life stress. So what we found in this study, and of course it's only in males, we saw that there was an association with perceived stress. So those who had the low activity variant have lower perceived stress, okay? Then we introduced a third component, which is looking at resilience. And what we saw, that resilience had an additive effect with MAOA on perceived stress, all right? Which was very interesting. And this was also published in PLOS ONE. So perceived stress is a broad measure of individual stress used worldwide, and the MAOA gene illustrates the influence of resilience to have an additive effect with MAOA. And that's why resilience is a significant factor to be considered when studying the genetics of mental health and stress. And this can only be discerned when you're looking at multiple time points, and therefore longitudinal designs are very important. And we know this is difficult under humanitarian conditions, but we were able to do it. This was published, as I said, in PLOS ONE. And this reminds us of the Orchid Hypothesis, where children especially, some of them are highly sensitive, that's why they're called orchids. Some are considered to be tough, and so more like dandelions and can adapt. And then we have the majority are tulips who lie in the middle. Again, this was work done Jordan and Yale University and University of Florida. Now, to tie in as the story is evolving, recently, in April of 2021, Michael Pelusi and others published a review in The Lancet talking about the role of epigenetics in psychological resilience. So now the needle is moving to look at not the negative impacts of trauma, but what are the positive aspects of it, and hence resilience and epigenetics. And in this paper, a theoretical model was proposed to outline the contribution of epigenetics in the development of psychological resilience. So as you can see here, this debate about nature and nurture is becoming very gray. So if we look here at the life course of a human being, first in utero and then born and then going until they're full adults, we see that we start with their genotype, the stable genetic effects that's part of the genetics, so that exists. But then we see here how the environment actually influences that in different stages of life. And we have both protective factors and negative factors, especially if there's adversity at any stage in the life of the human being. But also we have the epigenome, right? So what genes are being turned off or on, whether early in life or later. So there are ones that are stable epigenetic effects that always exist. And then we have those that are introduced because of the environment. Again, both in a protective way, pathways to resilience, or in a negative way, and how all that impacts our trajectory of mental health, okay? An outcome of resilience, which is a very complicated, multidimensional, let's say, description of this phenomenon. So again, this is recently we're talking about how all these factors combine together to impact the epigenome. So if we were to look at this, we could say that we have the genetics, which is the DNA sequence variations, but then we have the gene regulation, like what genes are turned on and off that form the phenotype. But what turns on and off a gene is not just the basic DNA sequence variation, but it's also the environment and how that, through epigenetic modifications. And so we need to always remember that when we talk about epigenetics, we're talking about persistent and heritable alterations in genome information that do not involve changes in the DNA sequence, right? It's the same genome, but multiple epigenomes because of the different effects of the environment. And so, of course, what does epigenetics mean? This is just to show you that at the molecular level, you have your DNA, and DNA is wrapped around proteins called histones. And if these histones are methylated through an influence from the environment, then you have some genes are turned off or on, meaning more tightly wound and therefore turned off, or loosely wound and therefore turned on. So these DNA methylation on those histones, the proteins that help fold the DNA, can either turn on or off a gene, right? And therefore influence function and behavior of a particular cell, which results in behavior or phenotype of a particular organism. So what kinds of exposure can influence epigenetics or influence the epigenome? So you have maternal exposure, meaning when the mother is pregnant, whether she's exposed to stress, nutrition, lifestyle factors, environment, trauma. Then you have and if she, through her maternal exposure, transfers that, then you have the fetus in utero is being impacted through the DNA methylation and the histone modification. And then we have the adult onset disease as a manifestation of those changes, whether manifested in the form of diabetes, hypertension, cardiovascular disease, etc., or obesity. So this is taking from Michael Kober's work on epigenetics, showing that you start, or you have, looking at early life, what are the disease risks that could happen to you over your lifespan, early life, midlife, and late life, and what are those factors that influence it, right? So you have epigenetic modifications that happen because of social determinants of health early in life or in midlife. And then you have directly social determinants that deal with race, ethnicity, economy, gender, lifestyles, education, and all that. So again, we're blurring this between what's nature and what's nurture. So this is work from more work from Michael Kober's lab at the University of British Columbia in Vancouver, where he has come up with let's say an epigenetic clock, specifically for children, a pediatric epigenetic clock, for taking DNA from cheek swabs. And what he has shown, right, is that this epigenetic clock that you can see here, the pediatric buccal epithelial clock, is impacted by a number of factors. And this clock kind of measures aging of a cell, right? By looking at the methylation that happens on the DNA. And so here you see intrinsic factors that impact this methylation and therefore the clock. And then we have physical and social environments that impact the clock. And then we could have interventions that help stop the acceleration of the clock or stop the negative impacts of whatever intrinsic factors or physical and social environments that happen. And all, the combination of all these factors is what results in the health and developmental outcomes for a human being. So working with Michael, we wanted to look at the impact of a particular intervention we have in Jordan, which is called We Love Reading. We Love Reading is a program that fosters love of reading among children by training adults to read aloud to the children. So it creates an environment of human-human interaction for fun. So again, it's a program that we hypothesized to reduce stress and anxiety and promote and boost resilience, as well as having a mindset of I can and a feeling of confidence and resilience, so to speak, and which impacts multiple sustainable development goals. And it's a very important thing, especially during COVID, with all the stress and anxiety that's been happening. We've already done research on the program to prove a few principles in the traditional sense. So with Dima Amso from Brown University, who's now in Columbia, we found that We Love Reading improves executive functions. We also found with the University of Chicago that it improves empathy. And so with Michael Kober, we wanted to ask the question of whether the pediatric epigenetic clock or the pediatric epigenetic aging can be, or whether it would be impacted by the We Love Reading intervention, especially among children who've undergone trauma of war, in this sense, refugee children or children from vulnerable communities. So the idea was to see what moderates the impact of the intervention, whether it's gender, socioeconomic level, mother-child relationship, and so on. And then to see if the social emotional development behavior can be impacted and changed, as well as taking into consideration any longitudinal sleeper intervention effects as well. So it's looking at all these factors. So in a nutshell, we wanted to ask, is there a difference in pediatric epigenetic aging acceleration between those that received the Wheel of Reading program and those that did not? So we're doing a randomized control trial. And then do pertinent individual factors such as gender, cognitive ability, and parenting moderate the potential association between the Wheel of Reading intervention and epigenetic age acceleration? Thirdly, does the epigenetic age acceleration correlate along the spectrum of social emotional development across individuals? And fourthly, is the potential association of Wheel of Reading intervention with the pediatric epigenetic age acceleration proximal, indicating immediate biological change as a result of the intervention, or distal, indicating potential intervention sleeper effects on in the longitudinal form? And to know if it were a proximal relationship, is that association maintained into a more distal time point? So the idea is to look at multiple time points, not just two. So here, this is the design that we would recruit the mothers and their children and randomly assign them to the intervention or not, and then for ethical reasons to give the intervention later on after we're done. And then introduce the intervention or not, and then so gather information at T1, pre-intervention, and then at T2, after the intervention, and then at T3, at least 6 or 7 months after post-intervention to look for distal effects. So this is ongoing, and so stay tuned until those results come out. But we also, to end the story, we also wanted to look beyond the lifetime of the individual and to look at inheritance of the epigenetic markers or patterns. We already know that in mammals, about 1% of genes escape epigenetic reprogramming through a process called imprinting. So we wanted to know, and we know this happens in mice, so we wanted to know what epigenetic markers escape in humans and are transferred or transmitted or inherited to the next generation. So this work has not been done in humans to prove a causal relationship. There are studies showing associations because it's not easy to discern whether the impact is because of the mother's exposure, the child's exposure in utero, or because the mother actually transferred those epigenetic markers. So what we're doing now is we have 3 groups of 3 generations of Syrian refugees, where in one group the grandmother was exposed to trauma, and so the hypothesis is that her daughter and then granddaughter would inherit the epigenetic signature of that trauma. And then the second group where the mother, not the grandmother, is exposed to trauma. And the third is where neither the grandmother or the mother or the daughter are exposed to trauma, and then to compare between them. And as my daughter says, she calls this the immortal kiss of our grandparents. And this is work that's been funded by the National Science Foundation, and we actually are analyzing the data as we speak. And this is, again, work with the University of Florida and Yale University. So I'll stop here, and I look forward to your questions. But this is an example of how curiosity, observation, and as the famous Nobel Prize winner in physics says, a scientist is somebody who sees what everybody sees, but thinks what no one has thought. Thank you very much.